Well, we are back, and we are going through our email backlog because, like, both of you are writing us a lot of emails. A lot of emails. Some of them are lengthy and complicated, and we want to actually answer them.
We could probably change the names of the people emailing the questions so it sounds like we got more people sending questions.
Let's not do that, though. Let's keep it honest.
So, right? Dave.
Oh, by the way, by introduction, the grumpy scientist, Mark Tommerdahl, Bob Dennis.
Right, and thanks for watching.
We have a fan, Dave S.
And he asked a lot of questions. I'm only going to answer one of them because they're hard.
Well,
I had to go.
Actually, responded by email in detail, but it's so sciencey.
Yeah, I know. I had to actually go back to my dissertation and was like, wow, I actually wrote
calcium channels
and
all that stuff.
and start reading that stuff. I was like, wow, I can't
believe
I wrote that.
Kind of thick. So what can we talk about that'll entertain people?
So basically, the question is about repetitive stimulation, and you kind of wonder, you guys don't do any repetitive stimulation, what are you talking about? Well, repetitive stimulation is actually the fact that one of the things that we take advantage of. Oh, I pre-drew the wrong thing, but here we are.
That's right.
This
is why we don't plan and script, because if we do so, it's always wrong.
Let's compare these stimuli. You know what these are, Bob?
Sine wave?
They're sine waves. And this is the shape of the stimulus that we deliver.
So this is like a little mechanical object moving up and down. Right?
Yeah.
In our case, at the fingertip.
Yeah, and so this is one reason it took so dang long to make the brain gauge, is because we deliver very precisely timed and precisely sized stimuli to the skin.
Much more precise vibration on your skin fingertip. If I may, a little plug for people who want to do this as a DIY thing. Brain gauge, probably not, because I believe our positional accuracy on
less than a
micron. So most people don't have an intuitive sense for a micron. All right, like a human hair might be tens of microns in diameter. An individual, maybe 100, 70 or 100. An individual cell in your body on average is 10 or 15 microns in diameter. So if you can imagine a cell in your body, okay, a blood cell, 7 microns, right? Mm-hmm. So we can do about a tenth of a blood cell in terms of the actual control of the movement of the mechanical vibration.
It actually matters too.
And it matters if you can't. That's the thing. If you can't do it that accurately, you can't measure the things we're measuring.
Yep, exactly.
Isn't it amazing that at your skin, with all that dry stuff and calluses and everything, you can still detect differences in the vibration that are 1/10 the size of a single red blood cell, and that is a fact.
And actually, it varies with frequency. So that's another thing that we control for.
Yeah.
And so if you deliver a really high frequency stimulus, and when I say really high frequency, most people automatically think, oh, you're going 5, 10, 50 kilohertz. And it's like, no, 200 hertz is really high
frequency for tactile stuff.
Tactile. So anything over 75 is considered high. Mm-hmm. And most of the stimuli that we deliver are 25 hertz. And basically what we're interested in, and what the question was about, was repetitive stimulation. So the thing that we studied for many years before we even started doing these studies with the brain gauge back in 2004 was, what's the difference between a short stimulus and a long stimulus? So when you give a short, and the quick answer, to those of you that want to bug out now, the quick answer is that a short stimulus gives a very broad, diffuse response in the cortex. In the brain, you get a very broad, it's like you activate everything, it goes, oh, okay, yeah, I felt it. Great. Then you start doing the stimulus over and over and over, and then you kind of shake around, your brain starts waking up. It's like, oh, okay, I'm feeling things. And
so this could be at 25 hertz.
You start developing a pattern
per second.
Yeah, exactly. And so what I drew here were a couple of groups of cells and theoretical columns. It's not a theory that columns exist, but my drawings are theoretical.
That's because they are chicken scratch. That's right. So they're mysterious and deep.
Yeah,
because they're hard to interpret.
So what this is 2 places in the brain that are side by side, and we're trying to imagine what the heck does repetitive stimulation do? Well, if we give, and this is a graph, X and Y, whatever. This axis here is time. Maybe I should have
time.
You try it. And this is
so you can make
this
fun
axis,
of me, but I won't do it.
Is intensity. Okay, over here we've got the same axis. Now let's say, same thing here, but this is really big. A is much greater than B. So basically we got two inputs. Now imagine I deliver a stimulus to the skin, just one stimulus. Some places in the brain are going to be a little more active than their nearest neighbor. And like I said, with a short stimulus, it's a who cares.
Yeah.
It's like, yeah, okay,
we saw it. Something there.
Well, the first thing that happens is it goes in, it wakes everybody up. And there's these things, and your cortex is made up of layers. There's 6 different layers, and the upper cortical layers have all these things called NMDA receptors. Well, NMDA receptors they're voltage sensitive, so that basically first stimulus doesn't do anything, but it does wake them up. And then the second stimulus, they're kind of going, yeah, this is looking interesting. And then the third stimulus, they're going, all right, yeah, we're going to start getting active.
So is it correct to say that they become more sensitive or receptive to repetitive stimuli?
They become much more sensitive, and they play a big role in repetitive stimuli. So when you give a stimulus that's about this big, and which is just a little bigger than stimulus B to the nearest neighbor, the response is going to be something like this. On the first response, there's just going to be a little bit of a difference. Can't really see much of a difference there, right?
Not the same.
Second stimulus, NMDA receptors are waking up. And so now that when they're waking up and they get turned on, first thing they do, they're the bullies. They're like, let's tell these guys, let's go over here and start turning this guy off. This is a little bit larger than this guy. So the next stimulus, because they're active, let's say on the second stimulus, they start getting active. It starts going up. And when they go up, they start driving this stuff down.
Do you remember the talks we gave on lateral inhibition? So these are points that are sort of adjacent on the cortex, right? And so if this one up here is getting a bigger stimulus, it will tend to suppress the response of its neighbors, correct?
Right. And what happens is basically it doesn't get much higher. After it gets sort of voltage gates get turned on and they get active, well, it's going to stay about the same. It's really going to stay about the same. But what does happen is the activity over here, even though there's only a little bitty difference, it starts getting down farther and farther and farther. So basically what you have is you have an exponential difference between the activity here and here, even though you only have a very subtle difference between those two.
Now, let me ask you a question, because I actually don't know the answer to this. I think I know the answer, but doesn't it even suppress adjacent areas down to below baseline activity?
Oh, yeah.
So it actually can kind of go negative. So like whatever the baseline sort of just hanging out activity is of your cortex, the area that's being stimulated can even suppress it below baseline. Even if it's not getting a stimulus, it can even be less active.
Exactly. And the whole point of this is to have maximum contrast.
Right.
And so and it's extremely efficient. So if you want to have maximum contrast, you can do a couple of things. You know, if in a non-biological world, you could just jack up the signal on one thing and just really crank it up and it'll go way above the noise. Or you could turn the noise down. So your signal-to-noise ratio gets better.
Or biologically, you do both.
Yeah.
Right. That's what the brain does. So the brain wants to sort of focus in and get maximum contrast. It sort of amplifies the thing that it wants to pay attention to, and it suppresses adjacent things that it wants to not focus on.
That's right. And so what we do with different tests is that we rely on the length of the stimulus. This is something that's very important. And I think we have a do-it-yourselfer that was wanting to write his own code, which that's fine if he wants to, but I think he was frustrated because the tests were too long. And he wanted to be able to test himself every 20 minutes or something.
It doesn't work that way.
Yeah. But basically, not only does, when you deliver, and when I say a long stimulus, 500 milliseconds.
Half a second.
Yeah. A lot of things happening.
Well, at 25 hertz, a half a second is going to give you 12 and a half cycles. And that's plenty of time for this to happen.
Right. Usually a lot of things happen in about 100 milliseconds. So, just short, we could do things like where we compare how do you do on a short stimulus versus a long stimulus. That's sort of where translational research comes in, because when you start using a really short stimulus, people go, it didn't work. I couldn't feel anything. Remember, we're talking about looking for really small variations. So what we do is we deliver 2 stimuli that are fairly long, side by side on both fingers, and if there's a really subtle difference, you'll be able to pick that up. So most people are seeing differences of about 10%, which is a difference of 20 microns on their DL. That's not a whole lot in terms of if you're trying to look at the tip of a pen or something like that.
Yeah, it's like about the diameter
small.
of a cell, of a living cell in your body.
So there's a lot of timing that's involved here. You've got the timing of how long the stimulus is. You've got the timing between the stimuli. And then, well, we have timing, and then you want a question-answer period. Well, that question-answer period has a minimum, so that if you don't let your cortex recover, and there's a fixed amount of time we think that it needs to recover, and that's the way it's set up, is that you're going to get bad results.
You get a bleed over from the previous test, is that right? So you've
That's
already
correct.
done some preconditioning on the brain. If you do another stimulus too fast, your brain is already sort of dynamically adjusted.
And this is actually one of the things that went on in the old. I mentioned in one of the videos how some of the reviewers were really grumpy about the way we were doing things, and the one reason they're really grumpy is because we developed ways to start stimulating 2 digits at once. And previously, most people would have to spend about $30,000, $40,000, $50,000 to stimulate 1 digit. And so what they would do is they would stimulate 1 digit. They deliver a frequency, a stimulus like this to one finger. They wait and they simulate again. They say which one was bigger.
Yeah.
But if you're doing it on the, you have to have all these elaborate protocols to figure out which of those 2 stimuli were bigger. Yeah. And most of those protocols took about 45 minutes, as opposed to one of ours that takes about 2.
Right.
So delivering, and so we take into account timing on different fingers as well as between repetitions. That's all taken into account. But if you want to do it yourself, knock yourself out.
Well, it's a really difficult thing to do. It's not like building a radio or something. It's more like building a satellite. So you can do it if you want, but you really have to if you're going to do this. This is one of the reasons you can't buy commodity grade stuff like a cell phone vibrator. Not the same, right? You can't control frequency and amplitude independently on those, and you certainly can't control it as well as you can control frequency and amplitude.
But somebody will do that, and somebody will say,
when you see the $22 brain gauge, clone, be skeptical because
it will be perfectly capable of doing what someone told us that the brain gauge could only be proven to do.
Yeah,
and that's
measure reaction time or something.
Or no, actually, it's even worse than that. We remember this was one of our potential customers said, my lawyer said I can't use this because you can only prove that it tests finger function. I'm going,
oh yeah,
oh
so,
wow,
so that's a threshold thing, right? Does it get through your calluses or anything like that?
It's
right?
A little bit like saying a reading comprehension test is just like. It's the same as testing your eyesight.
Yeah, reading comprehension and eyesight, they're related in some way, but you can cancel out the eyesight part of it and actually look at how your brain is functioning. Right?
Yeah,
so.
and if these test patterns bore you and all that, there actually are other test patterns, but we've got a lot of protocols, but we start everybody out on some pretty simple stuff.
Well, yeah, you can do more and more sophisticated, deeper and deeper sort of plunges into cortex function by doing different, more subtle things to your brain, because your brain is dynamically adapting to these stimuli, and so, or a healthy brain is anyway, right? So you can use the brain's response to something. Maybe half a second later, you can try something else, or a quarter of a second later, something like that. And so if you get your timing right, and your frequency right, and your amplitude right, and then something about how the brain talks to itself intracortically, you can really start to separate out some very interesting effects. And this is one way that it happens. So I think he was asking, is it exponential or?
Yeah, so this difference is exponential.
The difference, and that's sort of the focusing, right? Where it's your brain is focusing in on the area that's being stimulated and is suppressing all the areas around.
Right.
In its somatotopic map.
And it wouldn't be very evolutionary if it didn't. That's one way to think about these things. Is like, well, if it were linear, we'd have trouble navigating our environment. But we want to highlight the differences really quick. Right. And that's one reason we have set times for things and bore the cortex and then change it really quick. And that's what it's looking for, is those. Yep. And your brain's ability to adapt to these very subtle differences very quickly, dynamically,
is an evolutionarily driven, adaptation, because brains that don't adapt to their environment really quickly probably get eaten.
Yep.
Right.
Yep.
All right. Well, anyway, I think that's about it.
Thanks for watching.
Thanks for joining us, and I'm sure there'll be more.
You bet.
Please send us your questions. Bye-bye.